Hello everyone, welcome to Logic Medico. In this video, let's understand what is the basics of neurons, their interactions, their way of activation and inactivation. Starting from this, whenever a neuron, when a positively charged ion enters, especially the sodium ion entry, the inner side of the neuron will be more positively charged, and that process is called as depolarization. So usually it happens at the cell body. This level of depolarization has caused the generation of a nerve impulse, also called action potential. Also there will be subsequently efflux or exit of potassium ion from the neuron that results in repolarization, that is inside of the cell, because there is a loss of positive ion, inside of the cell becomes more negative when compared to outside, and this is called repolarization. The same thing is depicted in the form of a graph called as action potential. Here we can see RMP, meaning resting membrane potential, where the neuron is resting, there is no activity in the neuron. And there is something called threshold potential. The stimuli should be able to generate the nerve impulse. It has to reach the threshold potential, then only there will be generation of an action potential. So here we can see in the graph also I have shown the entry of positive charge ion like sodium resulting in depolarization or the upstroke, and there is a peak potential followed by potassium efflux resulting in inside of the cell becoming negative, that is called repolarization. And sometimes there is overcorrection, and that is called hyperpolarization, and back to resting membrane potential. This is the action potential of the nerve. Okay, so that is the basics of neuronal activation and inactivation, that is depolarization and repolarization. So what happens when the nerve impulse is generated? The nerve impulse ultimately reaches a button like area called as terminal button, also called synaptic button. When the nerve impulse reaches at that level, there will be opening of calcium channels. These calcium channels, when they open up, there will be influx of calcium as depicted in the diagram. The calcium ions enter into the terminal button. What happens in the terminal button? There are synaptic vesicles which contains the neurotransmitters. These calcium ions sticks to the outer surface of this synaptic vesicle and make them more sticky. When they become sticky, they attach to the cell membrane of the terminal button, and there is a exocytosis of the neurotransmitter. So the gist of this is, when the nerve impulse reaches the terminal button area, the calcium ion enters into that and makes the synaptic vesicle sticky, thereby causing exocytosis of neurotransmitter. So that's how the neurotransmitters are released. Once the neurotransmitter is released, based on whether they're excitatory or inhibitory neurotransmitter, two things can happen in the postsynaptic neuron. Either there is EPSP or there is IPSP. So what is this EPSP? Excitatory postsynaptic potential. The excitatory neurotransmitters released at the level of synapse travel across the synaptic cleft, act on the postsynaptic membrane, that is the next neuron, and they open up the sodium channel which are related to those receptor. When sodium channels are opened up, due to electrical gradient, sodium enters into the neuron, causing inside of the cell more positive than the outside, and that is causing excitation in the next neuron. Therefore it is called as EPSP, excitatory postsynaptic potential. This happens only in case of excitatory neurotransmitters, example glutamate and aspartate. Whereas in certain set of neurons, when there is a release of inhibitory neurotransmitter at the synapse, that is like glycine, gamma aminobutyric acid, dopamine, what happens at the synapse? Again there is opening of a channel, that is positively charged channel, but here due to the electrical gradient, the potassium ion will leak out of the cell. When the potassium ion leaks out of the cell, the inside of the cell becomes negative when compared to the outside. That is called hyperpolarization. GABA will act via a G protein coupled receptor and opens one channel called chloride channel. When chloride channel opens up, the chloride ion enters the neuron. When negative charge enters the neuron, again there will be inside of the cell is negative when compared to the outside. That is hyperpolarization or inhibition of the postsynaptic neuron. So this is called as IPSP, inhibitory postsynaptic potential. So when the excitatory neurotransmitter acts at the synapse, there will be excitatory postsynaptic potential, meaning the next set of neurons will get excited. When inhibitory neurotransmitters act at the synapse, the inside of the neuron becomes negative and by resulting in inhibitory postsynaptic potential. Also, you can just call it as depression of the next set of neurons. So what are these excitatory neurotransmitters and inhibitory neurotransmitters? Neurotransmitters are basically chemical substances which communicate between the neurons. So these excitatory neurotransmitters are stimulated to the next set of neurons. They can also be labeled as positive to the neuron. Glutamate and aspartate, they activate the neuronal activity in the brain. If glutamate and aspartate levels in your brain is high, you will not get sleep. Why? Your brain is too much charged up. The positively charged excitation is there, so you won't get sleep. Inhibitory neurotransmitters are the examples: glycine, simplest amino acid; GABA, gamma aminobutyric acid, and an amine called as dopamine. This inhibitory neurotransmitter causes negativity in the neurons of the brain, thereby causing hyperpolarization, thereby causing CNS depression. These are the examples: glycine, GABA, and dopamine. There are another set of neurons overall in our body is got abundant amount of acetylcholine, what I have written here as ACH. Most common neurotransmitter or the abundant neurotransmitter is acetylcholine. This acetylcholine causes excitation in the brain, whereas noradrenaline, on the other hand, the neurotransmitter of fear, fright, flight, sympathetic nervous system is noradrenaline. This noradrenaline causes inhibition in the brain because it causes negativity inside the neurons of the brain. Whereas a neurotransmitter like serotonin has dual action. At certain places it causes excitation, at certain places it causes inhibition. Actually, this is what happens. So always there is a balance between everything, that is excitatory neurotransmitters like glutamate, aspartate, acetylcholine, inhibitory neurotransmitters like glycine, GABA, dopamine, noradrenaline, and serotonin. So our brain is a balance of all these neurochemical substances called as neurotransmitters. Hope you understood the basics of neuronal activation and deactivation. It will help you in a long run to understand the drugs used in CNS. If you understand the basics of this, the drugs used in CNS becomes easy. So to tell the gist of that, any drug which prevents the entry of sodium ion prevents the depolarization. So when depolarization is prevented, action potential is prevented. Thereby, there will be CNS depression. Any drug which favors potassium exit favors repolarization. So when repolarization happens, more amount of repolarization, again CNS depression, because it's difficult to elicit or excite the neuron which is been repolarized. Whereas on the other hand, any drug which improves the sodium entry causes more depolarization. Causes more depolarization means more amount of action potential, so it can be used as a CNS stimulant. On the other hand, calcium entry, if you prevent the calcium entry into the terminal button, you prevent the release of neurotransmitters from the synaptic button, thereby you are preventing the neuronal transmission. So calcium channel blocker can be used to prevent the synaptic transmission either at the NMJ or at the neuronal level. And even the drug like ethosuximide, used as a absence seizure, used as a CNS depressant in a absence seizure, does the similar action. It prevents the entry of calcium ion into the terminal button, thereby preventing the release of neurotransmitters. When there are neurotransmitters, there is no excitation of the next set of neurons. This is how you can apply this basics of neuronal activation and inactivation. Thank you for watching and learning from Logic Medico. Kindly press the thumbs up icon if you like the video and had something to learn from it. Subscribe to our channel and kindly share this video with your friends too. Thank you once again.